Today we want to learn about meiosis. Meiosis is a quite organized anatomical event of which is quite a fascinating process that is taking place here. In school, of course, most learners, most students, they are struggling about this meiosis. What is actually happening here is where we are going to be learning about genetical variation, genetical diversity. We are going to be learning about cell division in most cases. So we have meiosis 1, we have meiosis 2. What is going to be taking place here is that we do have phases that falls under the category of this topic. Uh, Phase number 1, we have what we call is interphase. So I'm going to be drawing the cells that has to do with meiosis, and then I'm going to be explaining them on the side. The word interphase, what does it mean? This is where now another thing that you must consider is that interphase don't belong to meiosis, don't belong to mitosis. The word interphase. It is a process of which both of these topics begin with. Interphase don't belong to meiosis. Interphase don't belong also to mitosis. But we do need interface on both topics in order for us to have our cell division, because this is quite an important topic for us to begin with the word meiosis. What we are going to do here, we have our cell. Remember, this is interphase. If we have our cell like this, and then we're going to have our nuclear membrane, and then inside we're going to have our chromosome. So just a quick explanation, this will be a cell membrane. This is a cell membrane. Inside here, between this space and that space, is a cytoplasm. This particular layer here is a nuclear membrane. Inside nuclear membrane, this is a very dark staining area. It requires what we call is a light microscope to see this. This is microscopic. You cannot see it with your eye. This is normally a dark staining area, but we know one of the organelles that we find inside here is this chromosome. So here inside, you find what we call is nucleus. What is happening here, actually, I'm just giving you a short recap of a cell, what we are going to be learning about. So what does the rule of interface say? The first thing that is going to take place on this interface, let me give you another cell so that when I do the explanation, you can understand. The first rule of this, it says DNA must replicate. Duplication must take place. The word replication is just saying that make more DNA. So let's consider our lecturing today as this part, because this is the only part that we know. There are things that are carrying on here. There are organelles, there are centrioles, there are mitochondria, there are Golgi apparatus, there are a lot of cells that are in here. But let's focus now on the nucleus inside, where the DNA originates. Now, what does the first rule of interface say? The first rule of interface say, make more DNA, make replication of DNA. So let's see. They say, the first rule say, make more DNA, deoxyribonucleic acid. Make more DNA. That's the first thing that has to do with interface on the process of meiosis. What is the second thing? Actually, making more DNA is just same as saying replication. So now we understand here we must make more DNA. How do we go about making this? This is what we are going to be focusing, this chromosome. This hand and this hand, we call them chromatid. This in the middle, we call it is a centromere. This one and this one is the same thing. These are chromatids. They are divided by what we call is a centromere. And then when they are together, we call it is a chromosome. Chromosome. The word chromo means color. The word zome means body. These are colored body. You will see that in a minute now. So here they say make more DNA. It's quite important for us to make more DNA. And then it's quite important for us to understand how will this replication take place. Why is it important for us to make more DNA? But even if we understand that the DNA should be more, how are you replicating it? Where is it going to be more? Understand that we have this chromosome at the beginning as a diploid cell. Now we want to have, at the end of this, we want to have a haploid cell. So what is happening here? We have something like this at the beginning. This is a chromosome. But we know in interphase they say make more DNA. Replication must take place. Duplication must occur. How do I make this to be more? Am I going to write something like this on the side? No. I'm going to add one chromatid like this and then one chromatid like this. This is how you make DNA more. They say make more DNA. This is what you do. In another word, this means that DNA will produce identical copy of itself. DNA, deoxyribonucleic acid, will produce identical copy of itself. So sometimes what does this mean? This means that DNA is responsible to make a perfect copy of this chromosome. This arm and this arm are the same. This arm and this arm are the same. But the original arms were this one and this one. We need this for metaphase. We need this for anaphase. We need this for telophase. But again, let's see now. They say make more DNA. We understand that duplication has taken place, right? This is just the work of interphase. One thing that we shouldn't forget about interphase, just remember to make more DNA replication, and then DNA will produce identical copy of itself, meaning that this particular chromatid will produce this, and then this particular chromatid will produce this, but this is actually the same thing. And then when they are like this, they will change the name of chromosome to what we call is tetrad. Tetra for 4. When they are like this now, this is no longer a chromosome now. This is a tetrad. This is the process of making a zygote. This one. A tetrad means 4. Sometimes these two, you can call them chromatin sisters. Sometimes you can just call it chromatin. Doesn't matter really. So now we have finished our interphase. Let's go to our first step. Our first step will be, we know that we have prophase, but here it's quite important for us to write that is prophase 1. If it's prophase 1, on our interphase, if you can take, if you can focus here, you will understand that we don't have interphase 1 and interphase 2. This is just the process of interphase that can take place within existence. existence of meiosis and existence of mitosis. So we do have now the beginning of meiosis, things that are happening within meiosis. We have what we call is prophase one. In prophase one, we do have a cell. Going to write it for you. Remember, in interphase, duplication has taken place. Now, what is the rule? First thing that you must remember about prophase one, homologous pair come together. Homologous pair come together. One tetrad from the mother, one tetrad from the father, they will come together. First thing that you should remember. Homologous pair come together. Remember, in interphase, things were not together. There was another chromosome, there was another tetrad here, you know, there was another one here. So we need these things to really come together. Yeah, don't worry about these sizes. Tetrad are really different sizes. Homologous pair come together. Remember, this is happening inside the nucleus, but it will take place in early fetal life shortly after conception. This will take place, and then we do have meiosis that will be taking place within the existence of the testis, and then within the existence of the ovum, and then we have another meiosis that will be taking place shortly after the fetal life. Now they say homologous meaning that DNA that is coming from the mother and the father, meaning that tetrad from the mother and the father, homologous pair, two different DNA should come together. In this case, it will be from the mother and from the father. So what does that mean? This means that chromosome tetrad number one will start to intertwine with tetrad number one, meaning that they will be sitting here randomly, not necessarily that they will be lined up or somehow. They will be just here randomly. You see, this is what is happening. Now, homologous pair comes together, meaning that tetrad number one will pair with tetrad number one. Tetrad number two will pair with tetrad number two. Number three with tetrad number three. This is the mother, this is the father. So you understand, if they say homologous pair comes together, this is the pair that should take place. Tetrad number one from the mother should pair up with tetrad number one from the father. And then there is quite something that is also fascinating here. Let's look. We have duplicated our DNA, which is fairly okay. And then now we are going to be considering, after homologous pair comes together, what is the next step of this particular prophase one? Another step it will be saying now, um, crossover must take place. Crossover should, okay. This is the second step now. So now I'm going to draw a diagram of a crossover. I'm going to give you a diagram of a crossover. We're going to have Do you see? In crossover, this is where now these DNAs start to touch each other. This is a crossover process. Crossover process. This is where now this particular DNA from the mother and with DNA from the father, they start to cross the line, they start to touch each other. But we do find this particular topic also in our eye. As soon as the optic nerve has passed the blind spot, we're going to call it, it's going to reach a point of what we call is chiasma. Chiasma has to do with the crossover, but this now will be happening in your eye. But because here now we have 23 pairs from the mother, 23 pairs from the father, we're going to call this chiasmata because the crossover doesn't happen once. In an optic nerve, it happens only once. They cross over just before they reach hypothalamus. And then we understand that later on, it doesn't cross over again. But in this particular tetrad, we understand here it's going to be crossing over from the bottom and above. We call this chiasmata. This is also the process of crossover, but this is the process of crossover, but when it does crossing over more than one time, we call it chiasmata. When it's doing the crossover once, we call it chiasma. But the actual word that is quite fundamental for everyone to use is crossing over. This is when now these pairs will start to intertwine with each other. This is when now they will start to coming together, crossing over. Remember, for us to have this particular extra or identical copy of this DNA is fundamental for this particular crossing over process because this is where this arm and this arm they must get attached to each other. So now let me just find a space, a space. Now we have learned about interface, we have considered also prophase. Now we are going to what we call is metaphase. This happened under this topic of meiosis. This is metaphase. This is quite a difficult topic that students and learners come across. Meiosis is quite an organized anatomical event that has to do with physiology that cannot be seen with naked eye. This is quite microscopic, but it depends also with what kind of microscope are you going to use? If you are going to use a light microscope, it's also okay, but there will be organelles that will not be able to identify within the cytoplasm. If you're going to use what we call is electron microscope, that is where now you can actually see everything. This particular, the way the cell was invented by a guy called Robert Hooke. He's the guy who came up with the settings of a light and electron microscope to come and see what is happening inside the cell. But now let's focus in metaphase. This is also metaphase 1, right? Rule number 1. What is happening inside a metaphase? Anything that has to do with metaphase, you should remember this now. Metaphase contract things together so that they can be in the middle of the cell. Metaphase in whatever, whether it's meiosis, metaphase 1 or 2, they will make sure that the tetrads are in the middle. The duty of the metaphase make sure that tetrads are in the middle of the cell. I will tell you for what. Now, this is what is happening here. Uh, this must be contracted, lining, metaphase, line, tetrad, tetrad to the middle of the cell. How do you line tetrad to the middle of the cell? How is that even possible? That is quite possible also. Remember, we had in our prophase one, we had our tetrad, but they were not lined together. So now I'm going to demonstrate that using another colors. We have, now I want you to see this. This is our tetrad. This one will be the mother, this one will be the father. They are still doing the process of crossover, but soon we will have to pull them apart. In metaphase, we don't have really a lot of things that are taking place here. We know that this must take this to what we call is a metaphase plate. This process, we just call it is metaphase plate. This is all that is going to be taking place here. Remember also, these particular things that are carrying on here is just to show you that something must happen, but we don't know what is that. And then we don't know what is that can control the proteins inside in order for things to take place. In prophase 2, we did have the nuclear envelope, the nuclear membrane. But in metaphase 1, that particular membrane will dissolve, meaning that it will disappear, and then the lining will be like this. Now, we have passed this particular stage, we go into what we call is anaphase. Anaphase 1 also. In anaphase 1, what is it that is happening? We understand that metaphase land tetrad in the middle of the cell, we call that is metaphase plate. It will just line them in the middle of the cell. But in anaphase, what is it that is going to be happening? There is an organelle that exists within the cytoplasm. We call that organelle centriole that will develop here in the middle. Centrioles. Centrioles. But apart from these centrioles, the lining appears to be the same because they are still in the middle. Uh, Here we're going to say development of centrioles will take place. Development of centrioles will take place. That is number one. Number two, well, what will happen? If we have centrioles, we understand that there must be some sort of attachment to these centrioles. And then there must be a name for that also. We call it this microtubule protein, but in simplified term, we're going to call it this meiotic spindles. Meiotic spindle will also occur or will develop. What is this meiotic spindle? Why does we have to develop it here so that there can be some attachment? So that there can be some attachment. Good people, I'm sure you understand, all of you, what is the function of what we call the centromere. I'm sure you understand. Can you see we need to be holding on the centromere in order for us to enable the anaphase process to take place? There is nowhere else we can hold this without even holding this centromere. And also centromere help us to know how many chromosomes do we have, how many tetras do we have. We count them with centromeres. We can't count them one, two, three. We need to see the centromere in order for us to count them. So development of centrosome will take centrals will be here, and then meiotic spindle will develop. Meiotic spindles will be this tubule, this particular lining here. These are your meiotic spindles, and then this here, these are your centrosome. Now, the process is like this. What is the next thing? We understand if you're going to attach your fibers to something, yeah, in the knee we have tendons that is responsible to making the entire leg to be active. But in the cell, we have these particular spindles. Spindles are there to pull apart. This word anaphase, we know. When we will study anaphase 1 or 2, just remember that it will pull apart. It will pull this particular tetrad to different angles. And then metaphase, remember, metaphase will make sure that it line them in the middle of the cell, meaning that it will do what we call is a metaphase plate. Putting them in the middle, we call that is a metaphase plate. In anaphase, we know that this process must ensure that it will pull apart. Now let me give you an something to show you that how will those things be pulled apart. Because here they will be pulled apart. If they will be pulled apart, also the process is telling us that we are approaching the telophase. When you see anything dividing, because the entire topic is cell division, but when you see something dividing inside of the nucleus, remember that we are approaching telophase, because telophase cannot operate in a cell that is still like this. So what's going to be happening here? Let me just write those cells as they are. At the end of anaphase, this is what you're going to be having. This is what you're going to be having. They want to pull, the word pull apart, it means that this particular meiotic spindles must pull. These are like the suspensory ligaments that you find also in your eye that suspends the lens. But in this case, they will pull the tetrad apart. But you see, if it pull the tetrad apart, this is what is going to be happening. So what is the use of after pulling apart remaining with tetrad like that? There is really no use. But let me show you what will happen after this thing is pulled apart. We're going to be having things like this. You see, this is what we are going to be having. I just want to show you something. I should have used this color here actually. This one. I should have used this color. But colors don't really have that much impact when we do the teaching. Understanding has the impact. Now, in anaphase, the first step communicated with us that there was the process of crossover. Now, when pulling apart process take place, there are going to be genetical variation, D and A variation, and DNA diversity, meaning that DNA will be mixed by then. DNA will be mixed, will be mixed by then. This is now the process of this particular anaphase. In metaphase, the crossover as a result of prophase has taken place. And then here now we understand that because of this reason, when you pull them apart, DNA from this side will overlap to the maternal, maternal will overlap to paternal. This is why we have something like this. Let me just give you a clear diagram at the end of all this. When we speak about DNA verification, what are we speaking about? Just want to try to pull that up. I don't know if I will be able. This one. So we shall continue. This one will be like this. As a result of that. So here again, we're going to have something like this and something like this with something like this. Do you see the process now? There is no big difference between the mother and the father, but you can still see the original chromosome remain the same. Is this one on the side, but that one that was duplicated on the process of interface is the one that is doing this DNA verification variation, whereby they will be mixed together. We have DNA from the mother, DNA from the father, from homologous pair, they mixed now together. You see, this is the process of anaphase. Let's do the process of telophase now. And now we are going to be continuing. We ended up in anaphase. We know that now we must go to telophase. Telophase 1, because we have telophase 2. That's why I keep saying telophase 1, metaphase 1, anaphase 1, prophase 1. We have these things are duplicated. Now, in telophase, now you must remember now, in anaphase, we had the cell. The process of what we call is a pulling apart took place, and then the chromatids were on two sides of the cell, meaning that we must now do something like this. If now anaphase has pulled these things to be here, do you understand that in telophase, this was the process of anaphase. In telophase, this is where now we must start to develop something which is like this. This is the process. Now, again, the nuclear envelope will be reformed. The nuclear envelope will be reformed. But there is something interesting taking place here that divides these cells. This process, we call it the cytokinesis, that will divide these two cells into two cells. That will divide this one cell into two cells. We call it cyto has to do, kinesis has to do with movement. Now we have it like this now. Let me just do it the way we have it. We have it like this now. How do we have it again? We have it like this. And then this will be 23 pair from each cell. This was just the whole process of this to get the DNA variation, to mix just this DNA. Now, do you understand? If a cell is like this and a division must take place, this is a cytokinesis that will be taking place inside here. It will divide this cell like this, meaning that we'll end up with two cells as a result of this cytokinesis. But sometimes it can happen here. This will be 23 on one cell, 23 on the other cell. And the nuclear envelope will disappear again. These things are different size, don't worry. Now we have mixed DNA. We know that we have two different of a cell. This is now under the process of telophase 1. And then we know we have 23 from each cell, which is normal. 23 pair from each cell. 23 pair, 23 this side, 23 that side. This is how we end this one now. This is actually the last stage on telophase, on meiosis 1. We end it with telophase 1. So now we must go to the process of interphase again. But in interphase, there is no duplication of DNA that must take place. What does that tell us? That tells us that we don't need interphase in our second stage of meiosis. What we can do, we can just continue with our prophase 1. After this, we can have our prophase. We can have our prophase 2. So now, remember, just grab this particular concept. This is quite a little bit tricky, but be precise. Prophase, we know in prophase 1, duplication of those tetra took place. But here we don't need crossing over, we don't need duplication, but something must add up here again. What is it that must add up? We really don't know. But this is how something will add up. Let me show you what is that something. Remember now we are dealing with two cells. We are no longer talking about one cell. After stage one of meiosis, when we go to stage two, it will be our meiosis two. Here now, we don't need one cell. We are working with two cells because the process of DNA diversity has taken place. We do have mixed DNA now. We are ready to plant the zygote. So what will be happening here is that, okay, centrioles will develop. Centrioles will develop. This is what is going to be happening. And then it has never happened like this before. But can you see that something added up? Why is it this one happening now? I will show you now. I'm just going to draw one for just to simplify it. This is maternal, this is paternal, this is the mother, this is the father. It takes about 8 days of this process taking place. If you can understand the cell in biology, and then you're good to go. I don't think there's any other organ that can give you issues because now here you deal with things that you cannot see. This is microscopic. You can't see these things. You can't put it there and look at it. You deal with things that need microscope and even not even a light microscope. You need things that can do with electron microscope whereby you can see what is actually happening here. Now, this is what is happening now. Spindles will occur. Meiotic spindles will occur. Centrioles will develop. And then now, do you see in prophase to what is happening? From this particular cell, duplication that only took place, it was of these centrioles and this particular meiotic spindles. This that will must suspend this. So now let's go to metaphase. Don't ever forget the process and the work of metaphase. Metaphase is just there to line things, to put them in the middle. This is metaphase 2 also. This can never be one cell again. This can never do the crossover again. What can this do in the process of telophase? It will multiply to be 4 cells instead of this one multiplied to be 2 cells. And then at the end of meiosis stage 2, telophase will multiply this to be 4 cells. We don't do the crossing over again. That is done. So we know that in metaphase, things must be nice. In equatorial plate, things must be in what we call is metaphase plate. It doesn't really change this one. I'll just put these colors forever so that you can see what we are talking about. Mm. This will just line up. Metaphase will just take these things and line them together up. They are, but not like this. Like this. Yeah, like this. Because this is genetical diversity. Ne? In metaphase 2, things will line up together again. Tetrad line up in a metaphase plate. Ne? Metaphase plate. They will just do the lining up in the middle. The word plate, it means that they must be in the middle. They will line up in the middle. Just grab that metaphase, always line things in the middle. It doesn't change. And then quickly, let's go to anaphase. Anaphase 2. Here also we know that in anaphase 2 we know uh, that division must take place again now. Centrioles, centrioles, when you see centrioles, if you don't find them under an electron microscope where you'll be studying a cell, you must know that the meiotic or mitotic spindles must occur, but mitotic has to do with mitosis. But if we're studying meiosis, we know that these meiotic spindles will occur. So let me just, for the save, so that we can save time, I will just put one tetrad in each of the following. Let me just take this one. I will take this one because another one fell down, but it's actually the same thing. Let me rather just pause a little bit. This is our tetrad, and then we're going to have one. I'm just going to put one here so that we can save time, but actually there are 23 pairs. This is now haploid cell. It's no longer a diploid cell. This is haploid cell. Now in anaphase 2, things are lining up smooth and nice here. But if you see these centrioles, you must know that pulling apart will also occur. Pulling apart will occur. We put it here. Now, centrioles will develop. Meiotic spindles, so that meiotic spindles can pull apart, so that meiotic spindle can pull apart. In anaphase 2, we know that centrioles develop, and then meiotic spindles also develop so that it can pull apart. So this is just what is happening in anaphase. And then also again, there will be the process of cytokinesis here again. As we are about towards our end stage of which is telophase, it takes a while to get there, but finally here we are. Allow me just to put the telophase here because I don't have a visible space for you there at the bottom. Telophase 2. This is our final stage. Now, anaphase will pull them apart. If anaphase pull them apart, the process of what we call is the cytokinesis will take place. Cytokinesis will take place, and then we'll end up with four cells like this. Pulling apart will take place in anaphase 2 as a result of this particular cytokinesis. Ne? Cytokinesis. Cytokinesis is the line that will come and occur here in the middle, and then it will start pulling them apart. But now let's look. How will they look like on the telophase 2, which is our final stage? They will look like this. This actually can give you direction. You will understand. This will be like this. And then the second one, you look at this one. How does it look? This one is like this. You will see this now are chromosomes. This one now will be like this. And then another one, you look at like this one. Same applies to. Remember, these were those tetrads that we develop in interphase on our very first stage. This one. These two. These ones can never change, cannot change. For a matter of fact, let me just show you. This is our chromosome that cannot change. This one is the one that can change. So we're going to have it like this. You end up with 46 of this genetical variation. Do you see? After pulling them apart here, in telophase 2, they will be like this. And then here you must have 46 chromosomes. 46 chromosomes. Chromocolosomes body. 46 colored body. This will be the end of meiosis. On our next lecturing, we are going to be considering the fetal growth, and then the fetal line, together with the disease and the complications that we can find in the existence of a pregnancy. Thank you for this video.